Hi everyone, my name is Lexi and we're going to go over oxide nanoparticles. Alright, so we're going to look into a little bit of the history of nanotechnology. So nanotechnology is a branch of technology that deals with different dimensions and tolerances of less than 100 nanometers, especially the manipulation of individual atoms and molecules. Nanotechnology has several different industries and it's a very new and advanced area with the environmental sector and offers several advantages. As you can see, it has different diverse application sectors including biology, biotechnology, chemistry, medicine, pharmaceuticals, food and agriculture, the environment, electronics, and several other different industries, including the military. They also use a lot of nanotechnology. I'm a little bit more familiar with the military side of it, and it is actually very interesting to learn about how we're using nanotechnology to better advance our technologies. Nanomaterials are well described as nanoscale building blocks ranging from very tiny atoms and groups to complex nanoparticles with sizes ranging between 1 and 100 nanometers. Still very small. As you can see, there's different ranges on that photo to the right. Going up, it goes up in size, and it's very small molecules that we are working with. Nanotechnology provides these nanomaterials with different functionalities because besides improved selectivity and reactivity compared to their bulk counterpart materials because of their high density and high surface area to volume ratio, in addition to high reactivity. Okay, moving on, we're going to focus more on the oxide nanoparticles. So, structure of the actual oxide nanoparticles, there's classifications that can be identified for multiple nanomaterials, and that is size, shape, and morphology. Their classification is highly dependent on their morphology and psychochemical properties. When working with these different nanomaterials, various terminologies are often introduced that can be misleading to inexperienced scientists and newcomers to the field of research. So a clear understanding is needed before addressing the more critical issues related to nanotechnology because it is very new to multiple scientists and research. The structure of the actual metal oxide nanoparticles is based on a closed pack arrangement of oxygen anions, with metal cations occupying the sites in between. These are closed packed arrays, usually the face-centered cubic, the FCC, or hexagonal closed packed, and both can accommodate small metal cations and octahedral and tetrahedral entered in the sites. And you could see in the top picture that these are different types of nanoparticles that could be actually made, and this is the shape and the morphology that we were talking about. And in the bottom picture, this is what the oxygen with iron looks like in the nanoparticle realm. On the right side, you can see this is an actual picture pulled of what a nanoparticle actually looks like. Okay, we're going to move on to the properties of nanoparticles, and in particular the metal oxide nanoparticles. And we're going to, for metal oxide nanoparticles, they provide specific optical properties such as UV absorption, specific color absorption in visible regions, dichromism, and photoluminescence. So those are probably the main properties of oxide nanoparticles, and I did a little research into specific oxide nanoparticles, and the first one I found was iron oxide nanoparticles, and these have been found in wide applications in different fields of biomedicine. In the top right photo, you can see what the iron oxide nanoparticles actually look like in a nanoparticle scale, and it's very different from the bottom picture, which is the zinc oxide nanoparticles, which we'll get to in a little bit. Iron is a very important key microelement and plays an important role in the function of various different living systems of different hierarchies. Also, it is very abundant in its physiological functions, bring into question the ability of iron compounds at the same concentrations. On the other hand, to inhibit microbial growth and to positively affect mammalian cells. Presently, multiple studies have been published that show the actual antimicrobial effects of the iron oxide nanoparticles against gram-negative and gram-positive bacteria and fungi. Several studies have established that the iron oxide nanoparticles have a low toxicity to eukaryotic cells. It gives hope that the iron oxide nanoparticles can actually be considered potential antimicrobial agents of the new generation that combine antimicrobial action and high biocapability with human bodies. So now moving on to the zinc oxide nanoparticles. There are several unique chemical and physical properties, such as high chemical stability, high electrochemical coupling coefficient, the broad range of radiation absorption, and high photostability, which makes it among all the metal oxides a key technological material and has very wide application in various of fields. So now we're going to move on to the synthesis of these compounds. And I'm going to start off with the general methods that are used for synthesis of nanoparticles, of the oxide nanoparticles, and then we're going to go into what zinc and iron and how different those syntheses can be. So some of the general methods include the solution-based, vapor state, and biological. The choice of the synthesis method actually is determined by the psychochemical characteristics of the metal oxide nanoparticle, such as the size, dispersion, type of intrinsic or extrinsic and/or defects, morphology, and crystal structure. So moving on to the zinc oxide nanoparticles, these have several more different types of synthesis that are used, and they can be chemically synthesized using mechanical chemical processes, controlled precipitation, sol gel methods, vapor transport methods, solvothermal and hypothermal methods, and methods using emulsion and micro-emulsion environments. Moving on to the iron oxide, these are broken up into 3 different types of synthesis methods, which include physical, chemical, and biological. For the physical methods, these are very elaborate procedures which suffer from the inability to control the size of the particle and the nanometer range. So these physical methods are very hard to do compared to the other two. For the chemical preparation methods, these methods are very simple, tractable, and efficient, in which the size, composition, and even the shape of the nanoparticles can be managed. And these iron oxides can even be synthesized through co-precipitation of iron 2 plus and iron 3 plus by the addition of a base, which all in all, the size and shape and composition of iron nanoparticles synthesized through chemical methods depend on the type of salt used, iron 2 plus and iron 3 plus ratio, pH, and ionic strength. And as you can see, there's a little chart that kind of shows these different types of physical, chemical, and biological methods that is used for synthesis in the nanomaterials. So that shows the breakdown of what different syntheses we could actually use for specific nanoparticles. Okay, I'm going to talk a little bit about green synthesis. When I was doing this research, I actually stumbled upon green synthesis, and it is very interesting. I found this to be something that I really like doing because I like going green and being eco-friendly in a lot of things, and this is what caught my eye. So I'm going to go over how green synthesis is actually played in a role of the oxide nanoparticles. So to give a brief overview of regular synthesis, there's various amounts of physical and chemical synthesis approaches require radiation, high toxic reductants, and stabilizing agents, what could cause pernicious effects to human and marine life. So researchers found this different type of synthesis to actually counteract that, and it is called the green synthesis. So we're going to talk about what actually is the green synthesis method. So this green synthesis of metallic nanoparticles is a one-pot or single-step eco-friendly bioreductive reduction method that requires relatively low energy to initiate the reaction. This also includes cost-efficient procedures instead of very highly toxic chemicals that could be expensive. This is a more cost-effective synthesis. So now we ask why. Why does this matter? Why do we care about eco-friendly and bioreductive methods? So there are some problems with the regular synthesis, as I said earlier, that there's toxicity, but some of these synthesized nanomaterials are subject to the specific and actual applications. They could suffer from stability in hostile environments, lack of understanding in fundamental mechanisms and modeling factors, bioaccumulation and toxicity features, expansive analysis requirements, the need of skilled operators, problem in devices is assembling and structures, and finally recycling, reusing, regenerating all of these compounds instead of just using it once because it's so toxic to work with. So some benefits that we could see with the green synthesis are eco-friendly approach, as the toxic chemicals, as I said, are not used in the green synthesis. There is a reduction of the overall cost because the biological component itself acts as a reducing agent and a capping agent, so the overall cost of the synthesis process is reduced. Also, there is external experimental conditions like high energy and high pressure that are not required, which leads to energy saving processes, which again goes back to the eco-friendly method. And finally, the green synthesis allows a large-scale production of these nanoparticles, which saves you money and time, as many of these nanoparticles could be produced in a large scale. So now we ask what, like how? How does this work? How do we use different molecules to actually do the green synthesis? So we're going to focus on 3 main molecules, and that's bacteria, fungi, and plants. And I did more research on this, and there was yeast that could be used in various other eco-friendly things that could be used for the green synthesis. So we're going to focus on these 3, as these more attain to the oxide nanoparticles instead of just general nanoparticle synthesis. So we're going to talk a little bit about bacteria. So bacteria possesses the actual ability to reduce metal ions and are momentous candidates in nanoparticle preparation. For the preparation of metallic and other novel nanoparticles, a variety of bacteria species are utilized. Prokaryotic bacteria and actomycetes have been broadly employed for synthesizing metal and metal oxide nanoparticles. So these specific bacteria are used for metal oxide nanoparticles and are used during the green synthesis because the bacteria is able to reduce the metal ions to actually synthesize the reaction. So now moving on to fungi. So fungi-mediated biosynthesis of metal and metal oxide nanoparticles is also a very efficient process for the generation of monodispersed nanoparticles in well-defined morphologies. They act as a better biological agent for the preparation of metal and metal oxide nanoparticles due to the presence of a variety of intracellular enzymes. Competent fungi can synthesize larger amounts of nanoparticles compared to bacteria. So if we wanted more nanoparticles because we wanted a large scale of a reduction of something, fungi would be easier to synthesize because we want that larger scale or the nanoparticles, as to the bacteria does a little bit of a smaller scale. However, the fungi has many merits over other organisms due to the presence of enzymes, proteins, reducing components, and their cell surfaces. The probable mechanism for the formation of the metallic nanoparticles is excuse my pronunciation, enzymatic reduction, reductase. That's another word for the enzymatic reduction, reductase, in the cell wall or inside the fungal cell. Many fungal species are used to synthesize metal and metal oxide nanoparticles, as I said before, like silver, gold, titanium dioxide, and zinc oxide. So fungi is used for multiple different nanoparticles, not just the oxide part of nanoparticles. So now moving on to plants. biosynthesis techniques employing plant extract has been gained increased considerable as simple, efficient, cost-effective, and feasible methods, as well as an excellent alternative means to conventional preparation methods for nanoparticle production. So these plant extracts actually are used for synthesis, and you could use multiple different types of plants. Plants are everywhere. You could find any type of plant to do this synthesis, this green synthesis, and it's actually a very excellent alternate means to the preparation methods of a nanoparticle production, which I found very interesting when I was researching all of this green synthesis. There are various plants that can be utilized to reduce and stabilize the metallic nanoparticles in one pot synthesis process. Many researchers have employed green synthesis processes for preparation of metal and metal oxide nanoparticles via the plant leaf extracts to further explore their various applications. As you can see, I have some pictures of plants, and there is some process, if you follow the arrows, that you could follow along with, of how they actually use the plant to synthesize these green nanoparticles using green synthesis. Okay, so now we're going to move on to the actual applications of what is going on with the oxide nanoparticles. So there are multiple different applications, and metal oxides with sizes in the nanoscale range showed essential applications such as fluorescence, optical sensors, catalysts, photovoltaic, and biomedicine, and multiple other applications that are not listed here. Those were the most important ones that I found and picked out. Besides all these applications, nanomaterials have also been used like gas sensors and anode materials for fuel cell. And moving on to, I picked out an application for iron oxide nanoparticles. These are utilized in almost all the areas. Excuse me. Iron oxide-based nanoparticles are extensively utilized in almost all areas because of their unique magnetic, electrical, chemical, and optical properties. On your right, I have a picture of the zinc oxide nanoparticles and using fluorescence to actually find some kind of biomedicine component in the zinc oxide. And it's very interesting to see the nanoparticle scale of what this actually might look like. So I want to say thank you for listening to my presentation, and here are my resources.